Hello, welcome to another edition of CNA's Opus. And today what I'm going to tell you about is how to lose weight a CNA's way. How to lose weight a CNA's way. So first, let me start off by looking at, this is an adipocyte right here. So that's a fat cell. This is stored energy. And that's going to be propene 1, 2, 3 triol, which means it's going to have 3 carbons, and off the 3 carbons it's going to have an alcohol group, and off of the alcohol groups it's going to have a fatty acid. So it's going to be propane 1, 2, 3 triol as glycerol. So you could picture it like this. And there, 1, 2, 3, and those are going to be the fatty acids right there that your body's going to break down when it's in a euglycemic state. And the euglycemic state is what you would call like a fasting state. You haven't eaten about 8 to 10 hours, and that is in between 70 milligrams and 120 milligrams per deciliter of glucose in the blood. And when you reach that state, there's lipase enzymes that will start breaking down these fatty acids right here within the adipocytes. And the adipocytes, or the fat cells, only have insulin receptors. So if you go down here to my, see, like I'm an artist. I'm an artist too. So that right there is an insulin receptor on the liver, but the liver also has a glycogen receptor. So now we move down here to pancreas, and like the pancreas is like this, I don't know what I was drawing right there, but anyway, in the pancreas there's something called the islet of Langaniers, which is like I don't know, the next Harry Potter sequel. So, but anyway, that's what it sounds like anyway. But in there it has these clusters of cells, and it has these alpha cells and the beta cells, and the alpha cells are going to be making in a euglycemic state, it's going to be making glycogen, and in when the blood sugar goes up, in a fed state, the beta cells are going to start releasing insulin. They're going to start releasing insulin so that glucose is taken up into the cells. On the liver, see the over here that's skeletal muscle, and that's a fat cell, or an adipocyte. And what you're going to notice between skeletal cell and adipocytes is they only have an insulin receptor. I should have labeled that. That's an insulin receptor. They don't have glycogen receptors. So when you're in a euglycemic state, and the alpha cells are releasing glycogen, it only goes to the liver. It doesn't go to the fat cells, and it doesn't go to skeletal muscle, because they don't have glycogen receptors. And glycogen is, again, a 7-membrane spanning alpha helix, so it's a G protein coupled receptor. And insulin receptor is a receptor tyrosine kinase, and it's actually a pre-dimerized one. It's one of the more unusual ones. But if you look, the liver has an insulin receptor, fat cell has an insulin receptor, and skeletal muscle has an insulin receptor, but only the liver has a glycogen receptor. So when the pancreas at the islet of Langaniers down here starts releasing from its alpha cell starts releasing glycogen, it only goes to the liver. It doesn't go to fat cells, and it doesn't go to skeletal muscle. So the glycogen starts signaling to these downstream pathways here, so that the liver starts producing glycogen, and it starts, there's another cellular pathway where it's going to start breaking down amino acids into glucose. So there's 2 ways that the glycogen receptors are acting, it's gonna there's a polymer of glucose here, and it'll start breaking it down into monomers, and it'll start releasing that from the glycogen into the bloodstream. But it's also taking muscle here, and in a process of proteolysis, it's breaking that protein down in the muscle into the amino acids, and those amino acids are getting here to the liver, and those amino acids are getting converted from amino acids into glucose, certain amino acids anyway. And that's in a euglycemic state, so it's in a fasting state. And again, that's 70 milligrams to 120 milligrams per deciliter. So that is what's occurring, is glucose is being released via the breakdown of glycogen, and it's also breaking down protein that's within the skeletal muscle, and using those amino acids that are from the protein to produce glucose. So there's two ways. There's two ways that glucose is getting into the bloodstream via the liver when you're fasting. So you don't die. Another way is when insulin is not getting to the adipocyte or the fat cell. They have these lipase enzymes there that'll start breaking down the fatty acids that are there, and the two fatty acids that are found there is acetoacetic acid and beta hydroxybutyric acid, which you'll see right here. So that's acetoacetic acid, and the acetic acid portion, or the old name is ethanoic acid, is this portion right there. So there's your carboxylic acid, and there's a methylene group. So that's a fully saturated carboxylic acid, and that's ethanoic acid or acetic acid. And next to it is aceto right there, which is a carbon double bonded to an oxygen, and then a methyl group right there. So them together will when the fat cell is not receiving insulin, those lipase enzymes will start acting on it, and this will be released into the bloodstream, as well as the beta hydroxybutyric acid. So buta, that's 4 carbons right there. And the old way that they used to do it would be, instead of counting from the first carbon, 1, 2, 3, 4, the way that they used to do it was this would be the alpha and this would be the beta. So it's saying on the beta, there's a hydroxy group on the beta. So beta right here, there's your hydroxy group, the alcohol and the hydrogen right there off the beta. So it kind of it breaks it down right there. But again, it's the same thing. There's the ethanoic acid or acetic acid, and it's the same up here. The only difference is this one has off the third carbon in, this one has the hydroxy group, and this one has is double bonded to an oxygen right there. So that's the primary difference. But once this is released, this isn't soluble, so it bounds to something called albumin. It's a protein within the bloodstream, and the albumin will take it to the liver cells. Goddamn liver cells. It'll take it there via albumin, because it's a fatty acid, so it's not soluble. So it'll repel back in. It'll want to go back into the fat cell and stay with the fat and not go with the liquid that's within the bloodstream. So it'll bind on to a protein like albumin, and the albumin will take it to the liver. And then once it's in the bloodstream, it usually loses, well, it always loses this hydrogen right here. So once it loses this hydrogen right here, that hydrogen will bind to other things in the blood, and the blood will become more acidic. And this right here will turn into its conjugate base, which is, let's see, acetate. This will be acetoacetate, and this will be beta hydroxybutyrate. So they'll turn into their conjugate base while they're going to the liver, because they'll lose their hydrogen. But other things within the bloodstream will pick up this hydrogen. So then the blood will become more acidic. But you can't convert fatty acids into glucose, but the liver can convert them into ketone bodies. And the ketone bodies during a fasting state will go to things like the brain, keep the brain alive and well while you're not eating, again, so you don't die. But how do we lose weight here? Well, you really don't, because you'll probably die. But if you took out this insulin receptor right here, and you couldn't store fat in here, you'd have no fat, like you'd have no reserve. You wouldn't be able to make the ketone bodies. But if you got rid of this, there would be a lack of insulin signaling, and the insulin signaling is what inhibits the lipase enzymes. So with the lack of the insulin signaling, the lipase enzymes would continuously break down these the fat cells off of the glycerol molecule, and you make the ketone bodies. You probably become get ketoacidosis, which is not a good thing. But if you survive, you won't store any fat here anymore. Like, all this fat will be like chomped up there, but you won't store anymore, and you basically have to be living off of carbohydrates the rest of your life and glucose. Otherwise you die, but you'll lose weight. And that's the CNA's way of possible way to lose weight. Oh, and remember, this is important. This part's important. Only the skeletal muscle and the adipocytes have the insulin receptor, as were the liver cells or hepatocytes have glycogen receptors and insulin receptors, because the glycogen receptors and the insulin receptors have different roles. So once you're in a fed state, insulin will go to its receptor right here, and that'll shut down these glycogen levels will go down, number one. But number two, insulin will go here, and that'll start shutting off the intercellular pathways here that are breaking down glycogen and turning amino acids into glucose. So that's in there. Once the insulin is there, it'll start producing glycogen again. So glycogen will start being produced again, which is just polymers of the glucose molecules. And is that it? Oh yeah, when insulin is received over here, when insulin is received over at this receptor, this insulin receptor over here, this will start something called GLUT4, which is a glucose transporter. It's a vesicle that'll come to the cell membrane here, and the GLUT4 doesn't require any energy to consume glucose, but it's so that the skeletal muscles can create adenosine triphosphate, and skeletal muscle will then start making glycogen. But skeletal muscle, unlike the liver starts making glycogen. The skeletal muscle will also make glycogen, but skeletal muscle makes glycogen only for itself. In other words, when you're working out or exercising, which I know a lot of you are, the glycogen reserve in there when you're exercising is only being used for skeletal muscle. It's like a loop. It's only being used for itself. when you're low on blood sugar, the glycogen that's within the liver is released into the bloodstream for everybody to party. This is for everybody to party. This is for only skeletal muscle to party. So it's selfish and will only use the glycogen reserved for itself, but the liver is making glycogen for everybody. So I hope that's a little bit of an explanation. And I'm Rich, CNA. See you later.